Hi everyone, my teammates and I will be talking about stem cells in cartilage tissue engineering. And I will start by giving a brief introduction on what cartilage is and on what tissue engineering is. So cartilage is a tissue. It is avascular, a neural, and a lymphatic, meaning that it does not have blood vessels, nerves, or lymphatic vessels. And it is easily damaged, and the problem is that it cannot self-repair. But tissue engineering seems to be an encouraging approach in helping to regenerate cartilage. So it is a process which manipulates cells, scaffolds, and biological elements in order to develop functional tissues. And a requirement is that we must have a decent amount of cell sources, which is where stem cells features. So we'll be talking about the type of stem cells that can be used as a cell source for cartilage tissue engineering. The first is mesenchymal stem cells. So mesenchymal stem cells, they play a huge role in the repair of tissue and in the continuation of damaged cells. And they're able to move to a place of injury where they will prevent inflammation and bring about peripheral tolerance. Peripheral tolerance means that stopping the lymphocytes from creating an immune response from non-harmful materials. And they can be obtained from the bone marrow, from the peripheral blood, fatty tissues, and the Wharton jelly of the umbilical cord. So the Wharton jelly is a mucoid connective tissue, and it surrounds the two arteries and one vein of the umbilical cord. So the first mesenchymal stem cells is the bone marrow-derived mesenchymal stem cells. As the name suggests, they are derived or they are obtained from the bone marrow. And in a review by He Huang and others, they report that bone marrow stem cells were first used to repair cartilage injury, and the process was successful. And also report on bone marrow stem cells which were drawn from the iliac crest, which is like the area where the arching bones sit on the either side of the pelvis. And these stem cells were seated together with other cells on a hydrogel and in a collagen gel, and they were transplanted on a site of cartilage injury. And then after a few years, it was reported that there was a decrease in pain and there were no longer walking problems in the individual. And also in humans, autologous bone marrow stem cells, which means that these are stem cells that had obtained from the same individual on whom they are used. They were transplanted on an area where there was cartilage damage, and then after a few years, an MRI scan showed that the damage was fully repaired. Advantages of using the bone marrow-derived mesenchymal stem cells is that the cells don't need to be expanded or manipulated, but a disadvantage is that the quantity, meaning like the amount that can be obtained is not enough, or it's unsatisfactory. And the second type of mesenchymal stem cell is the adipose-derived stem cells. They are a promising cell source because they have high proliferation rate and chondrogenic capability, meaning that the ability to give rise to or form a cartilage. And they are formed from mesenchymal cells by means of successive transcriptional and non-transcriptional processes that take place throughout the human life. And in a study by Lin and others, they reported that human adipose-derived stem cells were obtained from liposuction waste, which is a process where they remove fat from individuals, and they were seated onto scaffolds, two different types of scaffolds, in order to create scaffold and adipose-derived stem cell structures. And in order to evaluate the chondrogenic differentiation of these stem cells, RNA was isolated. It was turned into cDNA in order to be able to measure the amount of cartilage-specific genes that were expressed using real-time PCR. And this diagram is the result of the RT-PCR. So we can see that the cartilage-specific genes whose expression was measured is collagen 2, aggrecan, and SOX9. So And the two types of scaffolds that were used, we had the WJ-PCR scaffold and the PCL only scaffold. And from this, we can see that the amount of these cartilage specific genes that were expressed, they were much higher in the WJ-PCL scaffold than in the PCL only scaffold. And here we can see that they use the GAPDH as a control. The GAPDH is a housekeeping gene. It is expressed in a lot of tissues in high amounts. And it was compared with collagen 2, and it was again proved that collagen 2 is expressed in much higher rates in the WJ-PCL scaffold than in the PCL only scaffold, while GAPDH was expressed in the same amount. And here it is the collagen protein, its content, again, it is high in the WJ-PCL scaffold and in the PCL only scaffold. And here we can see in the WJ-PCL scaffold, the cartilage formation is much more, it's higher, I can say, than in the PCL only scaffold. So again, from in this picture, we can see that the differentiation of adipose-derived stem cells, and it expressed high amounts of these cartilage-specific genes in the WJ-PCL scaffold. So from this, we can say that the type of scaffold which is used in cartilage tissue engineering is also affected, because it has to be able to interact in a good way with the stem cells so that they can be able to express the necessary genes. An advantage of this, of using the adipose-derived stem cells, is that they can be obtained from in large quantities from a less invasive procedure, because it is a procedure that's already happening and we are just taking them from the waste. And the last mesenchymal stem cell that I will talk about is the synovial-derived stem cells. They can be obtained from the human synovial fluid or the synovium of the knee and hip. And in a review again by Huang et al., they report that the synovial-derived stem cells, which were obtained from a horse, were implanted into a rat that had a cartilage defect. And the results on the rat showed that the cartilage damage was regenerated, and there was no immune reaction towards the foreign stem cells which were obtained from a horse. And an advantage of this, of using these synovial derived stem cells, is that they can be obtained with minimum invasiveness, and they have shown to be able to hold on to their great chondrogenic differentiation better than the other mesenchymal stem cells. So that's it for me, and my friends will continue to talk about the rest of the other mesenchymal stem cells and the other types of stem cells which are used as a cell source for cartilage tissue engineering. Hope you enjoyed this, and thank you.
Good morning, ladies and gentlemen. Today I will be presenting about the umbilical cord blood stem cells used in cartilage tissue engineering. As you all know that the umbilical cord blood is the blood that is left in the placenta or the umbilical cord after the delivery of the baby. And in cartilage tissue engineering, the umbilical cord is used as a source of stem cell for tissue regeneration, specifically that to cartilage repair and regeneration. And there was an experiment that was conducted whereby the human umbilical cord blood was combined with hydrogen, and they were used to test the cartilage repair in the mobile condyle innervator cord. In the experiment, 6 healthy male pigs weighing about 40 to 45 kg and aged approximately 1.5 years were used, and all of these pigs were obtained 1 week before the experiment and raised under the same environmental conditions. The experiment was performed with the available human umbilical cord blood line, and each line was used into many bits. During the experiment, the cartilage, the bone, the bone marrow were eliminated, and after the elimination, a mixture of human umbilical cord and 4% of HA hydrogen composites were transplanted into the area of the osteochondral defect in the right knee of each pig, which means the right knee was the experimental variable, and the osteochondral defect was treated in the same manner on the left knee of each pig and left untreated as a control, which means the left knee was used as a control. And once a day for the next 7 days, the antibiotic and intramuscular injection of a pain reliever were given to the pigs. And after a period of 12 weeks, the pigs were allowed to move the knee joint freely without any mobilization device, which means that the cartilage was regenerated and prepared. And as I said on my report, the umbilical cord blood showed many advantages when it is compared with other sources of stem cells that are derived from the mesenchyme stem cells, since the cord blood can be easily collected and stored after delivery of the baby without putting the mother and the baby in danger, and it is not necessary to perfectly mix them to the patient when you're using them. And they also contain cells that are normally found in the bone marrow. I'll also be discussing about the peripheral blood stem cell. The peripheral blood stem cell are mostly used in vitro studies, and there was a review by WLQ and others whereby which reports that there has been a study whereby the peripheral blood stem cell was used as a source of mesenchymal stem cell for articular cartilage repair. The goal was to determine whether the mesenchymal stem cell from the mobilized peripheral blood of New Zealand white rabbit have similar biological properties in vitro and chondrogenesis in vitro as the bone marrow derived from the mesenchymal stem cells. And the results showed that there was no difference between the peripheral blood derived from the mesenchymal stem cells and bone marrow derived from the mesenchymal stem cells was found regarding their morphology, the immune phenotype, and anti-apoptic capacity. And the bone marrow showed to have a greater osteogenic potential and proliferation capability than the peripheral blood. The peripheral blood had a greater potential for adipogenic and chondrogenic differentiation than the bone marrow derived from the mesenchymal stem cells in it. Thank you all.
Good day, ladies and gentlemen. My name is Mukherjee Sote, and I'm going to proceed from the embryonic stem cells. Embryonic stem cells are derived from the undifferentiated inner mass cells of the human embryo. Embryonic stem cells are pluripotent, meaning they are able to grow into all derivatives of the three primary germ layers, namely the ectoderm, the endoderm, and the mesoderm. Furthermore, they are derived from the embryos during the developmental stage, just before the implantation occurs in the uterus. Human embryonic stem cells differentiate into the chondrocyte lineage because cartilage tissue engineering is the differentiation method for embryonic stem cells of the mouse. The chondrogenic differentiation method is the best method that can be perfectly used for human embryonic stem cells. Now, moving on to the process of the method of the chondrogenic differentiation. Human embryonic stem cells and iPSCs were differentiated into chondrocytes using micromesh culture. Briefly, embryonic stem cells and iPSCs were cultured at high density, very, very high density, approximately 1 multiplied by 10 to the power of 5 cells per 10 milliliters times 7 spots in 6 centimeters cell culture for straight 3 hours. Then after incubation, medium was added to each dish without dissociating the cell drop. For differentiating pluripotent stem cells into chondrocytes, the differentiation medium contained the DM, EM, the non-essential amino acid, the penicillin, and so on and so forth. So now moving on to this part, which is the results. The human embryonic stem cells and the iPSCs differentiate into chondrocyte lineage. As it has been demonstrated before, the cartilage tissue engineering is a differentiation method for mouse. So in this study, it is modified that this method for human pluripotent stem cells. For chondrogenic differentiation, high cell density microcell culture was treated with low concentration of FBS, insulin transferase, selenium, etc., etc. Moreover, the formation of aggregate starts to occur over there. Moving on to this part over here. The human embryonic stem cells form the cartilage-like tissues in vitro and in vivo. What does that mean? Following micromesh chondrogenic differentiation, human embryonic stem cells formed aggregate and expressed cartilage matrix protein lichen as detected by alkaline blue staining and collagen 2 COL2 as it's shown over there in figure A and figure B. Then differentiated embryonic stem cells expressed chondrocyte-related genes, which is the SOX9 and the COL2, as shown in figure C over there. Then following 6 weeks of culture, human embryonic stem cells formed their phenotypic hallmarks of cartilage as indicated by histological and immunohistochemical COL2 staining, which is the figure D over there. And the transplantation of aggregates, as you can see, forms over in figure E. Now moving on to the last part, which is the discussion and conclusion. Though the timing of tumor and dying tissues observation, we couldn't clarify 3 types of abnormal differentiation, namely the early or late tumor differentiation or formation and the cell death. In the early tumor formation close, we observed tumors following 6 weeks of differentiation. We couldn't identify any abnormalities in the late tumor forming lines at 6 weeks. Then these tumors usually occurred 2 weeks later. However, if we treated differentiation cultures with the p53 inhibitor, peripheral tumors, they could have occurred a little bit much earlier. Why? Because the peripherin increased the penetrance of tumor formation, and normally this is used daily just to increase the sensitivity of our interrogation, making sure that we didn't miss the iPS clones that were mildly tumorigenic. Thank you very much.